DNA replication. In this lecture, we discuss this point, the models of DNA, DNA synthesis and elongation, DNA polymerases, the enzymes used in the process of DNA synthesis, origin and initiation of DNA replication, the prokaryote and eukaryote models, telomeres replicating the ends of chromosomes and its importance. DNA replication is a process by which a cell copies its DNA prior to cell division. This process ensures that each new cell receives a complete set of genetic information. During DNA replication, the double-stranded DNA molecule is unwound and separated into individual strands. Each strand then serves as a template for the synthesis of a new complementary strand by the enzyme DNA polymerase. As a result, two new DNA molecules are created. DNA replication is an essential process for life, and any error can lead to mutations that could be detrimental. This process is referred to as semi-conservative replication because each of the original DNA strands serves as a template for one of the newly synthesized strands. The models, the semi-conservative and conservative and dispersive model. The equilibrium density gradient centrifugation, this is the experiment DNA in cesium chloride and centrifuge for 50 to 60 hours at 100 ground result in generation of gradient of cesium chloride and the binding of DNA. The increasing density in this way. In 1958, semi-conservative model of DNA replication, and this is experiment. In 1955, Arthur Korenberg worked with E. coli, discovered the mechanism of DNA synthesis. A Nobel Prize in Physiology and Medicine in 1959 was awarded jointly to Serge Ochau and Arthur Korenberg for their discoveries of the mechanisms in biological synthesis of ribonucleic acid, RNA, and desoxyribonucleic acid DNA. Ochau was a Spanish biochemist, while Korenberg was an American biochemist. Arthur Korenberg was American, who was awarded Nobel Prize in 1959 for his discovery of the mechanisms in the biological synthesis of 6-ribonucleic acid, DNA polymerase, an enzyme which is used for all kinds of recombinant DNA technique and for sequencing. Korenberg joined Sergio Ochau laboratory in September 1953 and worked on the synthesis of DNA after learning in 1955 that Ochau and his colleague had created synthetic RNA. In E. coli, he worked with E. coli. 4 components required: the desoxyribonucleoside, triphosphate, the sugar bases, a DNA template, DNA polymerases, which was called Korenberg enzyme. After that, DNA polymerase 2 and 3, discovered soon after. Magnesium optimized DNA polymerase activity. Serge Ochau was a pioneer researcher in the field of biochemistry and enzymology, and he is known for his work on the synthesis of RNA and the enzymatic mechanism involved in the process. In December 1955, he, along with his colleague, made the First discovery of enzyme DNA polymerase. Arthur Colenbeck was born on March in 30 of March 1918 and passed away on October 26, 2007. The main feature of DNA synthesis reaction: DNA polymerase 1 catalyzes formation of phosphodiester bond between 3 OH of the desoxyribose on the last nucleotide and the 5-phosphate on the D and TP. Energy for this reaction is derived from the release of 2 of the 3-phosphate. DNA polymerase 1 finds the correct complementary double-stranded DNTP at each step in the lengthening process. Direction of synthesis is 5 to 3. This is DNA elongation polymerase. DNA elongation is steps formation of phosphodiester bond, incoming desoxyribonucleoside triphosphate in this way, and this is the mechanism of elongation. Not all polymerases are the same. There is difference nucleotide from the 3 end of the chain. Origin of replication. A prokaryotic example begin with the double helix denaturing into single strand, thus exposing the bases. Exposes replication bubble from which replication proceed in both directions. Progression to 2 replication fork moving in the opposite direction. The replication fork. Segment of single-strand DNA are called template strand. Gyrase, a type of topoisomerase, relax the supercoiled DNA. Initiator protein and DNA helicase bind to DNA at the replication fork and untwist the DNA using energy derived from adenosine triphosphate. DNA primase next binds to helicase producing complex called primosome. Primase synthesize a short RNA primer. Polymerase 3 add nucleotide. The RNA primer is removed and replaced with DNA. Single-stranded DNA bind protein. This is the model of replication of E. coli. Initiator protein, DNA helicase. Helicase load onto DNA. Helicase denature the helix. And here rhizome formed. Primase synthesize RNA primer, which is extended as DNA change. The DNA replication is continuous on the leading strand and semi-continuous on the lagging strand. Unwinding of any single DNA replication fork proceed in one direction. The 2 DNA strands are of opposite polarity and DNA polymerase on the synthesis of DNA 5 to 3. DNA is made of opposite direction on each template. Leading strand synthesized from 46 in the direction of the replication fork move. Lagging strand synthesized from 5 to 3 in the opposite direction. Semi-continuous require many RNA primers. The supercoiled DNA relaxed by gyrase, unwound by helicase and protein. Okazaki fragment. RNA primer replaced by polymerase 1 and gap is sealed by ligase. DNA ligase seals the gap between Okazaki fragment with phosphodiester bond. DNA ligase the gap sealed. Initiation of the RNA primer made by DNA primase. Start replication of lagging strand. Synthesis the first oxide fragment. Polymerase 3 single-strand DNA binding protein. Further untwisting and elongation of new DNA strand. Polymerase 3 dissociates to continuous synthesis on this strand. The process continues, and second Coxsackie fragment finished. The third being synthesized, primer removed by DNA polymerase 1, joining of the adjacent DNA fragment by DNA ligase. Some concept we use: gyrase and helicase required. This is question for discussion. Why are gyrase and helicase required? The difference between template and primer, primase and polymerase replication 4. Why are single-stranded binding protein required? Leading strand and lagging strand. The replication of circular DNA in E. coli. 2 replication fork result in theta-like structure. As a strand separate positive supercoil form elsewhere in the molecule. Topoisomerase relieve tension in the supercoil, allow the DNA to continue to separate. A rolling circle model of DNA replication common in several bacteriophage, begin with the neck at the origin of the replication. 5 end of the molecule is duplicated and act as a primer. Polymerization at the 3 end. Attachment of the ribosome and the formation of Coxsackie fragment. During viral assembly, the DNA is cut into individual viral chromosomes. DNA replication in eukaryote. Copying each eukaryote chromosome during the S phase of the cell cycle presents some challenges. Major checkpoints in the system: cells must be large enough and the environment favorable. Cell will not enter the mitotic phase unless all the DNA has replicated. Chromosome also must be attached to the mitotic spindle for mitosis to complete. Checkpoint in the system include protein called cyclin and enzyme called cyclin-dependent kinases. Eukaryote enzyme: 5 DNA polymerases from mammals: polymerase alpha, beta, polymerase gamma, polymerase delta, epsilon, and other for repair. Each eukaryotic chromosome is one linear DNA double helix, average about 10⁸ base per long, with a replication rate of 2 kilobase per minute. Replicating one human chromosome would require about 35 days. Solution: DNA replication initiate at many different sites simultaneously. What about the index or telomerase of the linear chromosome? DNA polymerase ligase can't fill gap at the end of the chromosome after RNA primer is removed. Big problem. Its gap is not filled. Chromosomes would become shorter each round of replication. The solution is the recruitment. Eukaryotes have randomly repeated sequences at the ends of their chromosome. Telomerase composed of protein and RNA complementary to the telomere repeat bind to the terminal telomere repeat and catalyze the fission of the new repeats. Compensate by lengthening the chromosome. Absence of mutation of telomerase activity results in chromosome shortening and limited cell division.